Okay, so in this tutorial, we will predict the structural ensemble of an amyloid protein, which is predominantly disordered, that's amyloid beta 42, by using the CABS Flex 2 web server for coarse-grained simulation of protein motions. So molecular dynamic simulations predict the behavior of a complex system, physical movements of atoms and molecules especially and their interactions. So it generates a trajectory of motions of all atoms in a protein which evolves over time. However, in case of IDPs like the amyloid beta 42, the system that we will be using in this tutorial, the simulation time of evolved structures in the form of trajectory would not be an ideal representation due to the inherent fluctuating native states of these IDPs. That is, the lack of fixed or well-defined or ordered 3D structure. So the best representation of the dynamics of IDPs would be in the form of structural ensemble, from which the average properties will be calculated and benchmarked against the bulk experiment. So that's the standard procedure now for IDPs. That is, you generate an ensemble of structure either by molecular dynamic simulation or by using this particular server or by any other method. So there are a number of different methods. There's Monte Carlo simulation, there's replica exchange, or there's advanced sampling methods. So you can use all those methods to generate an ensemble of structures as if they were evolving over time, but they're not actually. So they could be anywhere in the time state of the evolution of the IDPs, let's say. And the dynamics will be defined by that ensemble, whereas the properties will be calculated as an average of that ensemble, which could be compared with the experimental bulk values, especially the NMR chemical shifts and J coupling constants and paramagnetic resonance enhancement, as well as SAX data for that matter. So CABS Flex 2 can run these simulations by reduced all atomic representation, as in like coarse graining the all atomic representation. That is, groups of atoms could be considered as a bead, and that allows it to simulate these IDPs at a faster pace, or say in a reasonable amount of time, as opposed to running traditional molecular dynamics simulations where you would require a longer period of simulation time in order for the properties, like most of the properties, to converge or reach a Plato state, let's say. There are other methods as well, like enhanced Monte Carlo types of simulations methods or replica exchange simulations or metadynamics, which could be performed. So these are advanced simulation methods that could be performed with intrinsically disordered proteins. But one way to go about it is by running the coarse graining simulations. So once it runs these coarse graining simulations, it generates a trajectory containing about 1,000 frames or 1,000 different structures in the coarse-grained representation, and from there on it does a clustering method to bring down the number of structures to 10, let's say. So there'll be 10 different groups or clusters which will have the representative structures of those 1,000 different initial structures that would be distributed based on certain criteria, let's say. And then from there on, it would rebuild the all atomic representation of those 10 different clusters, that is, taking a representative structure from each of those clusters and build back the all atomic representation in order to give you 10 different models. So that's how you get 10 different models by this CABS Flex 2 web server. So this is the website address, which I have posted in the slides as well. But before that, CABS Flex 2 requires an input structure. So the input structure, in our case, since we are dealing with a natively unstructured protein or IDP, that is amyloid beta 42, so we'll have to have a structure which is completely unfolded in that regard. So amyloid beta 42, 1 to 42 residues completely unfolded, and from there on, CABS Flex will generate a ensemble of structures which might have certain structural propensities transiently from certain regions within the protein, let's say. But we don't know about that. So we will be basically starting with a completely disordered structure. So in order to generate the disordered structure as an input to this CABS Flex 2 server, so I'm just going to click on this submit button, and then it will show you where to input the data. But before that, we'll need to generate a reasonable disordered structure for amyloid beta 42. So one way of going about it is this Nmr Utility website. So this is the server as well, which actually takes in a structure or a sequence for that matter, as in the primary sequence of the protein containing the amino acids, one letter code. and it can generate the structure. Once you upload the file in here, it can generate the structure in the disordered extended state. So that could be used as an input for the CABS Flex 2 server there. So this is a very good utility that's being developed and maintained by the BACS group, who are one of the pioneers in nuclear magnetic resonance, or NMR spectroscopy, especially for IDPs. But then again, in order to have an input to this server, that is, you can have a sequence input as well as a structural input. I prefer to have a structural input actually. So in order to have that structural input for this server particularly, you need a structure first of all, but there's no structure for amyloid beta 42. The only structure, or a few of them, are available in the protein data bank, as you can see with the PDB ID 1IYT. So you might wonder that since these are intrinsically disordered proteins, that is, the native state of these proteins should be disordered completely, but this is forming some kind of a helical structure throughout. The reason being, although this is calculated by NMR, the solution NMR, the solution here is not water itself, but a combination of an organic solvent and water, which in this case is hexafluoroisopropanol. And the percentage of water used in this solution alongside hexafluoroisopropanol is very, very less. It's actually not correct to state that the structure is solved in water, but we will use this structure anyway. So once you solve the structure for the IDPs in any other solvent that could actually retain the structure or give you a static pose, of course by NMR these structures couldn't be solved in X-ray crystallography because it's too difficult to obtain a electron density map with the structures because of those high temperature values, Therefore, these structures are always solved in NMR, and even in NMR it's difficult to obtain a solid structure. But once you have it solved in an organic solvent, then it has some propensity to have a structure for itself, which in this case is the helical structure that you can see, which is also the kind of structure that you would get to see when this amyloid beta 42 binds to a membrane environment, let's say. Okay, so we'll go ahead and download this PDB file. As you can see, there are a number of models in this PDB file, so we will select just one model from here. So we go to download files and we download the file in the PDB format, and I'll just save it inside my work directory here, and the ID for the PDB is 1IYT, so I save it here. And when I view the structure in Chimera, you'll see that it is an ensemble of structures. So there are actually how many? Maybe 10 different models, but the representative model out here is model number 1. So we'll go ahead, open the structure in text format, that is in the TXT with GVim, and just select model 1 from there and keep it, but I'll delete rest of the other models altogether. So if I open this structure in GVim again, and if I scroll down further, you'll find something called representative. So you see here the best representative conformer in this ensemble is null, which means they cannot really detect a single conformer that might be best representative. So amongst all the 10 conformations that have been submitted in the PDB file, any one of the conformers could be selected as the best representative structure. So there is no one particular preference that the experimentalists denote here as the best representative conformer. So we'll scroll down and just select model 1. So you see on top here, all the models are annotated with model 1 and then model 2 here, model 3, 4, 5, 6, 7, 8, 9, and 10. So we are going to delete all the rest models 2 to 10 and just keep model 1 there. Okay, so that's the beginning of model 2 here, and until model 10 we'll delete all the other models. There you go. So we just have model 1 here now. So we can check that actually by opening the structure again in Chimera. There you go, just one of the models. So we'll just submit this model of the helical conformation of amyloid beta 42 into the Nmr utility, that is by the BACS group, the web server, and ask it to generate an extended random coil structure, which will be further used as an input for the CABS Flex 2. So we go back to the BACS group and we just choose the file here, single model save. That's by the same file name because we didn't change the file name here. So it's overwritten on top of the downloaded NMR structure. Okay, now here you need to additionally uncheck this option because as you can see here, it states reset backbone angles to helix, which you don't want. So the backbone angles should be as a random coil conformation in order for it to be disordered in nature. So we'll just go ahead and uncheck this option. And then you need to have your email ID at this place as well. So the academic email ID, that is your UL ID that you have. So you need to confirm it once again and just hit submit. So after a while, there you go. In the next page, it will show that the calculation has been submitted to the queue, and that's your job ID, and the results will be sent to this particular email ID that I've given for the job to be complete. So if you go back to your email ID and just check if you have received an email for the job completion, you will find that your job is complete out here, and the email will be from spin.niddk.nih.gov. So it's from the National Institute of Health. At the bottom, it will actually attach the structure of the PDB, and it will have the name as ext.pdb. So just go ahead and save this attachment. That's this one, workspace directory. Now save the structure here and just go back to the terminal. So if I type in Chimera ext.pdb, you'll find the extended structure, random coil structure of amyloid beta 42 being generated by the Nmr utility. So you see all the helical regions are gone, and it's just plain and simple random coil structure with no apparent or visible secondary structure elements here. So we're going to go ahead and use this structure as an input to the CABS Flex 2 web server for generating our ensemble of 10 different models. Now, before uploading this extended random coil structure of amyloid beta 42 into the CABS Flex 2 web server, we'll have to annotate the chain IDs because the extended structure that was generated doesn't have the chain IDs automatically annotated. So we'll annotate a chain ID A here because the CABS Flex server always wants a specific chain ID. So you open the file in the text format again, and as you can see, the chain ID field here is empty. So we'll go back to Chimera, open the structure again in Chimera. So that's ext.pdb, and then go to Tools and Structure Editing, and then you have Change Chain IDs. So Tools, Structure Editing, and Change Chain IDs, and just click on that, and it will ask you to select the structure, the principal chain that you have for the PDB. That's ext.pdb out here, and since it doesn't have a chain ID, so it doesn't show any alphabet out here. So we'll change this to A. We'll annotate the chain ID as A. So just press OK, and then you have this new structure. Then go to File, and then Save PDB. Give it a new name. Let's say we give it a name of ext underscore chain dot pdb, and we'll locate to your workspace directory, which is this one out here, and just save it. Use the untransformed coordinates again. So save this one and then close this. And once again, we'll open the newly generated PDB file in the text format, and we'll find that the chain IDs are annotated now. So you have the chain ID as A in the missing field that was there. Okay. So now we are going to have this ext underscore chain dot pdb as an input structure or input confirmation of the random coil to the CABS Flex 2 web server. Okay. So just go to your browser again, and you have this CABS Flex page open, which we already did before while we hit the submit button here. So you browse to locate your file, and a new window pops up asking you to input the structure, the input structure that you will have for the random coil that we just generated there. So that's ext underscore chain dot pdb. So we'll go ahead and select that and open the structure. So you find this option chains out here, for which you have to input A, which is the chain ID that we annotated, that particular monomer of amyloid beta 42. random coil structure. You can give a project name to it. Let's say we give it a name of AB42. That's the short form for amyloid beta. And then you give an email ID. So email ID is necessary out here because this job, since this is a simulation, this might take a while to run. So it will give you the results in a separate results page in your, send you to the email ID actually. So you have an additional option saying do not show my job on the results page, which you can opt for, but it's not required. Also, you have a few customizable distance restraints, which a user can define by using this distance restraint generator. So you could bias your simulations to a particular secondary structure by defining these restraints. So mode tells the program to keep the restraints for at least one or both distance restraints. So basically, if you select just one residue, you have to choose SS1, and if you get to select two residues, you have to choose SS2 for that matter. And for both of these, the secondary structure will be defined based on the particular distance restraint that you have defined, so be it helix or sheet, whereas mode all will basically define the restraints for all residues, and mode none will not define restraint for any residues. Similarly, the option gap that you have here, it sets the minimum number of residues that should be skipped in a peptide when defining restraints between two residues. So let's say residue number 17 in the amyloid beta peptide, which is the part of central hydrophobic domain, cannot be restrained together with numbers from, that is residue numbers from 14 to 20, which means that in a gap of 3 residues on either side of residue number 17, that is starting from 14 to 17 and then from 17 to 20, that part of the residues will not be restrained alongside 3, whereas the other residues will be. Similarly, you have a feature where you can define the minimum and the maximum distances for the restraints itself. That is, these are measured in angstrom. So CABS Flex 2 has this default value of 3.8 for the minimum distance, whereas 8 for the maximum distance here. There's another option for the rigidity of the models. So the rigidity of the protein can be assigned in 3 different ways. So firstly, by providing number between 0 and 1. So 0 will stand for complete flexible regions. That is, in case of IDPs, it applies mostly in case of IDPs, whereas rigidity value of 1 will denote it as completely rigid. That is, it will have some secondary structural features. Now, between these two values, you can specifically define the rigidity values between 0 and 1. Let's say rigidity of 0.5 will have best of both worlds, like you will have a bit of flexibility as well as a bit of rigid regions as well. Okay, so the second way to define the rigidity would be by using the BF or the B factor, that is the temperature factor that you have. As you know already that in the X-ray crystal structure, if you're using an X-ray structure, then certain column will have this B factor values. So based on those B factor values, it will define the specific rigidity of the residues. And thirdly, you could also provide a custom file with one rigidity number per residue, which should be uploaded out here. So using the format residue ID followed by the rigidity. So the smaller rigidity value of the two restraint residues that you provide here will be taken into account as the restraint strength. Okay. So finally, you have another option to manually edit the C alpha distance restraints by selecting this option from the menu whether we want to edit or not. Additionally, you can also provide C alpha distance restraints and side chain distance restraints. So by annotating certain global weights to those restraints in both cases, in case of C alpha distance restraint, which is basically the backbone distance restraints and the side chain distance restraints of the protein. And on top of that, like before, you can also upload the restraint file if you want in both cases, in case of the C alpha and the side chain distance restraints, in the form of the B factor, as well as having certain values between 0 and 1 for each of those residues. All right. So we will not set any restraint for the simulation. That is, we will not put a bias on it. This will be an unbiased, free de novo simulation here. So we'll not set any values for this. So finally, you have options to specify parameters to control the coarse-grained simulations. So first, the number of cycles. So the total number of cycles are the total number of models to be saved in the trajectory. That is, a default of 50 cycles would mean 20 times that number is 1000 models will be saved in the coarse-grained trajectory. Further, cycles between trajectory frames will set the number of models skipped while saving the actual models. So cycles between frames of 50 will skip every 50 models, and this will also affect the total number of generated models, which will now be 20 by 50 by 50. So 20 times 50 by 50, which is equal to 50,000. So the final model number will be 1000 because the total number of models generated here would be 50,000, and if you're skipping every 50 frames, that would give rise to total number of 1000 models. Then we have a value for the temperature of the simulation. So as you can see from the values here, this is a dimensionless reduced temperature value that closely corresponds to the actual temperature. So ideally, one would have a temperature of 310 Kelvin as the physiological temperature for performing the simulations generally, if you're performing the simulations in case of the biological system, like in this case, these proteins are found in the brain. So a T of 1 would mean that the structure is close to the native state of the protein, that is the state where it should be functional enough, which in this case is actually intrinsically disordered, whereas a value of 2 would mean typically enables the structure to completely unfold. Now, since we have an unfolded structure already as input, a value of 1.4 would essentially not bias complete unfolding, but at the same time would also retain secondary structure properties in regions where it was supposed to retain. So the reduced temperature representation has the same meaning as a product of the Boltzmann constant and the real temperature, but it's more reasonable, or let's say it's more friendly to have a value between 1 and 2 as the temperature of the simulation, so that it could be precisely set in order to mimic the conditions that you want to simulate your system on. Finally, we have the seed for random number generator, which is just a random number that will be used to run the simulation. It's like an annotation for that particular simulation. So if you want to repeat the same simulation, you can go ahead and set the same random number that was generated out here, like 8763, with the same input models and everything, and it will try to generate as closely as possible to the previous simulation to the previous trajectory of models that it generated. Okay. So all said and done, we are not gonna change these default values and keep it as is. So ideally, it would first generate 1000 different models, like coarse-grained models, and from there on, it will cluster and produce 10 different models, which will be further converted to the all-atomistic representation. So you would finally end up getting 10 models. That is the ensemble for this IDP here. So we are going to go ahead and hit submit to run the simulations. So once we do that, it might take a while again, depending upon the busyness of the server and everything. So there you go. It goes into the status mode, stating that it's still pending, and it will be placed in queue at some point of time. And so it gives you the date of the job started and the time of the job started and the estimated finish time and everything else. As well as it gives you an option to download again the input structure. It actually derives the sequence from the structure that you have submitted as an input. And as we expected, it cannot annotate the secondary structure because it's a random coil structure of the IDP. All right, so you'll see this page keeping refreshing all the time in order to proceed with the job. And finally, you will get a result in your email ID actually. So this is the file if you have for C alpha distance restraints and the protein rigidity and the protein restraints and everything else that is submitted as an input out here. So the temperature range again is 1.4 and 1.4. So okay, so this simulation does not actually use a solvent. So this is basically an implicit solvent simulation. That is, it's not an explicit solvent simulation. So it regards the water implicitly, or let's say the effect of the water is treated implicitly out here. So we'll just wait for this job to finish, and once it's done, it will send you an email and it'll let you know when the job is finished with a link, and then you can click on that link and get to the results page to see and download the models there. All right. So the job is done now, and as you can see, the status is showing as done here, and you'll get the results in the model section that's here. Okay, so if you go to models, it will represent all the models as an ensemble. So 10 models here and it is represented as an ensemble, which you can also view with this inbuilt viewer that they have, the CABS Flex 2 server. So as you can see, it's a jungle. So it's basically spaghetti with little bit of secondary structures, which are mostly beta sheets, or let's say beta strands. So in case of monomer, since this is a monomer, the beta strand will fold it on itself, another beta strand, and it will basically give a representation of a beta sheet, but it's actually called a beta hairpin here. So let's go through each of these models one by one. Let's say we view the first model here only. So you'll see it's almost a spaghetti with a little bit of fold here and there. So these are basically the N terminal and the C terminal regions which are folded upon itself. And then the model 2, model 3, model 4. So model 4 is completely disordered, as you can see, although here a little bit a helical propensity is seen here in this region. Model 5, 6, 7, 8, 9, and 10. So we're going to go ahead and download all of these models together, okay? So we select model all again and hit download in the workspace directory. So let's say it saves it as multi-model out here. So you save the PDB file, and if you open that in Chimera, it will basically generate your ensemble of structures, which you can actually separate from each other by downloading the individual structures here and create basically 10 different files for 10 of the models. Alright, so that's it about coarse-grained modeling or coarse-grained simulation for generating a structural ensemble of a typical IDP, that is amylide beta 42, by using the CABS Flex 2 server.